So I guess first I'll thank the organizers for giving me the opportunity to present today. And when Hatice reached out to me to present, she asked me to kind of talk specifically about different spatial transcriptomics technologies. And I have a little bit of work from my lab on some of the biological applications that we've started using, just to point out an illustration, although we just had a wonderful illustration from Yufei. But I also wanted to mention that in addition to running a research program, I also am the director of the Next Gen Sequencing Core here at the University of Pittsburgh, the Health Sciences Sequencing Core at Children's Hospital. So we've been doing spatial transcriptomics for about 2 years now in the context of the core facility. So if those of you who are local, if you have an interest in doing spatial, please contact the core and we can help you get those projects set up. All right, let's see if I can advance the slides. Is there a trick? How do you advance the slides, Yufei? No, I don't.
Oh, sorry. Okay, cool.
Nice. Okay, great. Super. Yeah. Yeah, I'm just going to try and get the pointer to work. There we go. Oh, beautiful. Okay, thanks. Sorry for your thanks for your patience. Okay, so Yufei get a nice introduction to some of the imaging-based technologies. So I'm going to actually take a step back and break it down one step further. So there's really kind of two different categories of spatial transcriptomics technologies on the market currently. And there's also many sort of non-commercially available versions of these, certainly that came out to start with. But I am going to focus on the ones that are commercially available. So you can basically break them down into either a sequencing-based spatial, which means that you are doing a little bit more of an unbiased technology where you're trying to capture the whole transcriptome, or you have the imaging-based. Yufei just talked about some of the imaging-based, so I may not spend as much time on those as I go forward. And so there are key things to be thinking about with each of these two technologies, right? So the sequencing-based, and I'm going to go through them in a little bit more detail, but the sequencing-based technologies in general are aiming for as close to unbiased or an unbiased capture of the transcriptome. They typically are using either barcoded glass slides, beads, or nanoballs. The resolution of these technologies really varies tremendously with the different application. Right now on the market, currently we have everything from 2 microns in resolution all the way to 55 or larger microns. These are NGS sequencing-based outputs, right? So they're all going to go into a sequencing machine and you're going to get back reads that you have to align. And the imaging of the tissue is done either simultaneously or serially in order to locate your cells of interest related to the spatial data that you get back, the sequencing-based data. In contrast, the imaging-based technologies are using, in general, a targeted selection of transcripts using pre-designed probe panels, right? And that's where there's a lot of space in the field, going from a small number of probes, like 200, now getting up to 5,000. The nice thing about the imaging-based technologies is, although you lose that unbiased nature, you get much better resolution. So these are almost always at the subcellular, single cell and subcellular level of resolution because you're able to detect individual transcripts, right? These are a microscope output. It's an imaging-based output. So there is no sequencer. There is no sequencing. And the segmentation algorithms are needed then to locate your cells, which cell is actually there in the context of the imaging of the transcript, right? So really different kind of approaches that are needed computationally when thinking about the technologies, each of these technologies. Okay, so I'm going to go through the sequencing-based types first, and then I'll do a little bit on the imaging. And I'll try to highlight some of the things that maybe Yufei didn't have time to discuss in more detail. Okay, so all of the sequencing-based methodologies tend to fall into sort of a variety of categories. They're usually these sort of glass slides that have printed onto them or covalently linked onto them barcoded regions, right? So each of the regions is individually barcoded, and that's where that resolution can really scale depending on which barcode is at which region. That's the way that spatial is done for 10x genomics, and I'll go through that in a little bit more detail. There's also SlideSeq, which does have a commercially available application that I'll talk about briefly as well. That uses nano beads, which are individually barcoded, and those are then placed arrayed onto a glass slide. There are also some versions of this that are less commercially available, such as StereoSeq or PixelSeq or SeqScope. There's also a microfluidics version called dbitSeq. Broadly speaking, though, each of these are creating landscapes in a space on a slide with individually barcoded regions, right? And so it's really the resolution that can be different from technology to technology, depending on whether they're able to fit a variety of different barcodes into different regions of space. So to sort of get at that point more directly, the original version of the 10x Visium spatial gene expression that was released in 2022 had basically these capture areas. Each capture area is sufficient for one sort of tissue. The capture area is 6.5 millimeters by 6.5 millimeters. And within that 6.5 millimeter space is 5,000 individually barcoded circles. Each circle is 55 microns in diameter, and there's a 100 micron distance center to center between two individually barcoded spots, right? So what that means, first of all, is that there's space between the spots with no barcode, and you're going to lose information there. And also it means that 55 microns is a pretty big size for a cell. So most cells on average are in the like 5 micron to 20 micron range. Some of the bigger cells can get up to more than that. Most of the cells are going to be probably closer to 10 microns. So 55 microns is going to encompass definitely more than a single cell for most tissues. Also, thinking about tissue density can matter here. If you have a lot of cells that are really packed into a very small space, you're certainly going to lose information in between each of those spots, as well as have a lot of information captured on one spot. The actual barcodes itself, in addition to having the spatial barcode and a partial read that's necessary for the downstream sequencing applications, also has a UMI, which is of course important to ultimately track back to how many individual unique molecules you were able to capture of a distinct transcript, as well as a poly-DT tail. That's how it's trying to capture the RNA, right? So it's going to capture all of your poly-A RNA. Now, 10x Genomics just released, and I mean like a month ago, just released their HD solution to their resolution problems. This gets at single cell resolution. Basically, now you have the same capture area size, 6.5 by 6.5, but now you have a grid instead of circles. And on top of, in that grid, they have arrayed individual barcodes down to the 2 micron space, right? 2 microns is actually usually smaller than a cell, so you're getting almost at the subcellular resolution, really. Because you actually have to start thinking about how many transcripts can I capture in an individual space, especially when you're talking about just a piece of one cell, you actually start to lose, you have a kind of lose your numbers of genes, right? So it becomes a sparsity of data issue. So at that point, what they've decided to do is to actually categorize these 2x2 bins into an 8x8 larger size bin. And so when they initially sort of process the data through their pipeline, it comes back to you in these 8x8 bins. Now, an 8 micron bin is probably about the size of one cell. And then if you want to go in further, you can go down to the 2 micron resolution within those 8 micron bins. I think that's actually kind of a nice way to do this system so that you get that level of resolution you want, but you kind of have a slightly zoomed out view at that initial processing stage so that you have a little bit more data per cell. Now, remember, of course, if you have a dense tissue, those cells don't beautifully lie down on those 8 micron bins, right? A lot of those bins are going to have half of a cell and half of a cell. So what is the workflow for all of these? So basically the workflow is generally speaking a situation where you have to start with your tissue in a block, you have to cut it, and you have to cut it onto these capture areas, right? Once you've cut it onto the capture area, you can image it. You can either do H&E, which is sort of a basic pathology staining, gives you sort of nice regions, or if you really want to get down to the level of individual proteins, you can do immunofluorescence. Most of the standard assays are thinking about standard immunofluorescence, like maybe 4 colors. Of course, those of you who are doing much more complex spatial proteomics might want to think about assays where you could scale up and have much more proteomics. And I think that's something that we're looking at on the horizons of the future very soon. Once you've stained and imaged, you permeabilize the tissue. And at that point, the RNA sort of floats gently down out of the tissue and sticks onto those poly-DT tails. And that is how it is captured. You do an on-slide cDNA synthesis step, And then from there, you can actually pull all of the cDNA off of the samples. They've all been now individually barcoded, as well as each sample is individually barcoded in that synthesis step. You can pool them together into one tube to make your libraries, do your amplification. Off to the sequencer you go, and then you have your data. So when thinking about the landscape of tissues out there in the world, I think it's not unobvious to think about all of those medical pathology samples that have been biobanked for many, many years. And the reality is that for most pathology samples, the method used is not a fresh frozen situation. It's generally FFPE or formalin fixed paraffin embedded, right? So it became pretty clear pretty early on to these companies that they were going to need to create a system to capture FFPE. What I just described to you is essentially a fresh frozen workflow where you have decent quality RNA in your sample that it's going to float out during that permeabilization step and stick to those barcoded slides. So in order to get around the process of an FFPE, the main challenge being that FFPE really degrades RNA. That's like the first thing it does. Was to basically start by making pre-designed whole transcriptome probe libraries, okay? And this is actually now something that 10x has started using in their single cell platforms. They have their new assays called Flex, which you can use on a fixed sample, and that's basically the same concept. Let's design probe libraries that are specific basically to the entire transcriptome. So now what you can do is take a degraded RNA sample, you can hybridize your probe libraries to that slide, and then rather than you do like this on-slide ligation, then you permeabilize from there, and what you're pulling out is those ligated probes, and those probes are then sticking to the poly A tail, to the poly DT tails that are on those barcoded slides, okay? So now it's sort of in this very strange space that's sort of unbiased, right? It's unbiased in the sense that it is whole transcriptome, but it's not entirely unbiased because, of course, you have actually designed it according to what you think are the transcripts that exist, right? Just something to keep in mind. The other key problem with an FFPE solution like this is you've designed it to a specific species. So these are currently only available for human and mouse. If you want to do non-human primate or something off those two species, you're kind of out of luck. Okay, so I also wanted to briefly introduce the SlideSeq assay, which uses these 10 micron nanobeads. It's essentially the same process as before. One of the key problems with the current commercial applications through Curio, SlideSeq, or Tile, this is Curio Biosciences solution to spatial transcriptomics. You cannot image the same tissue that you're taking the sample from to do your transcriptomics. This is also only available for fresh frozen. They do not have an FFPE. Now, of course, they were sort of ahead in getting down to that resolution. Now that Visium HD is out, it's unclear to me exactly what the relationship will be between these two technologies and how utilized they will be. However, I will say that there's one application here that I think if you are not in a situation where you need to do FFPE can really be beneficial, and that's thinking about TCR and BCR sequencing, right? So for those of you who are interested in immune cells, T cells and B cells, each of those cells have individual T cell receptors and B cell receptors that actually function like individual barcodes for those cells. And when they get activated, they clonally expand, and you can actually determine whether a cell has seen its antigen and expanded in a tissue space and its relationship to each other using those TCRs and using those BCRs. And so a group at the Broad Institute basically introduced a version of SlideSeq where you can do a separate sequencing run by taking your cDNA library and then designing probes to the TCRs and BCRs. Again, this would be species specific. And now you can actually do a separate run where you're doing a TCR sequencing assay, and you can incorporate that information into your spatial data. And so that's shown here from this paper that was published in Immunity a couple of years ago. And this is looking at renal cell carcinoma metastasis to the lung. And you can see very nicely, they can both identify tumor as well as lung tissue and a boundary area using the spatial portion of the transcriptomics data, but then they can also go in and look at the T cell receptors and identify different TCRs that have clonally expanded and have different spatial distributions either in the tumor or outside of the tumor, right? And that can tell you something about tumor infiltrating T cells that might have some interesting use for your data. Okay. Another sequencing-based application that actually was probably the first on the market, but is probably going to be utilized a little bit less going forward, is something called the NanoString DSP Geomics. This is a system where you can actually select regions of interest to pull out either barcoded antibody or barcoded transcripts, probes And then initially this was sort of set up to be a little bit of a small scale probe-based situation with an N counter, but they rapidly transitioned it to a whole transcriptome probe library that they generated, and then you could do that on an NGS technology. Again, this is not single cell, but what is kind of nice about this platform is if you actually are interested in comparing very specific morphological regions that you can identify with protein staining, you can kind of go into your tissue and select those specifically and then just sequence those regions. So maybe you're interested in specific regions that you see within a tumor versus non-tumor tissue. This sort of technology gives you the option of staining with your proteins, really looking closely at the tissue of interest and selecting regions of interest that you want to go after at the whole transcriptome level and do spatial transcriptomics that way. So just another option and something to keep in mind that I think for some people makes more sense for their biological question. So of course, lots of different options. The question becomes, how do we compare them? The question I often get is, which one is the best one? Please don't ask me that question. So it depends. It depends on what you're trying to do. And that's what I'm hopefully trying to convey. But I think there are some key things to be thinking about with quality control. One is the capture sensitivity per area unit, right? So this is highly dependent on the tissue type. And I think what we've learned in our core facility, having done this on over 10 different human tissues and over 7 different mouse tissues, is that it really depends on the tissue. Some tissues are very transcriptomically active and they're happy to give you their RNA. Some tissues are a little bit more sparse. There's a lot of fat content. They're not as transcriptionally active, and you can get a lot of variation across your landscape of even the number of transcripts per region. Obviously, the spatial accuracy of the mRNA detection is something I think, especially when you're looking at fresh frozen tissue where it sort of gently permeabilizes and falls out, is something to really consider. These initial technologies were considered in terms of lateral diffusion issues. They don't seem to be a lot of lateral diffusion, but I think with a very dense tissue that's very transcriptomically active, it's hard to imagine there isn't going to be some diffusion process and you're going to get some sort of mixing across these barcodes. Sequencing efficiency is always something to consider, and the spatial area that you've covered is also something to consider. I'm not sure that benchmarking across these platforms with consistent tissues has really thoroughly been done. There have been a couple that have been done. I've referenced one here. Ultimately, how do you actually validate these things? Rigorous validation with a SM-FISH type assay to define tissues with known genes of interest probably still is something that needs to be done and is not done very often for these types of technologies. Okay, so that's all I have about sequencing. I'm going to move quickly through the imaging-based stuff. Yufei already kind of talked about this, but essentially, again, all of these are using this process by which you use pre-designed probes. Different companies use different methods of detection of those probes, and but ultimately, all of them are coming at this from a space where they're just going over and over again, doing cycling, multiple rounds of cycling to detect these fluorescent markers, right? So they're able to basically use combinatorial multiplexing so that each mRNA has a very specific combination of fluorescent images over multiple rounds that they can decode with their machine in order to tell you which transcript is where. And so the first one that came out was MERFISH. This was developed also at the Broad Institute. This is a really cool technique that uses a combinatorial barcoding assay. It's quite mathematically complex and does have some issues with molecular crowding once you start adding more and more barcodes. There is a commercial application of this called MERScope. This is just an image kind of giving you the subcellular resolution that you can see. So you can see one transcript in the nucleus, one transcript that's both in the nucleus and in the cytoplasm, and one that's even sort of more dense within both of those tissues. So you really can get beautiful, beautiful level of resolution with these technologies. This is the commercial application of this. It's the MERScope. The company does require that each individual person design their own gene panel. And so it's really, there are no pre-designed panels available. They expect you to go in, design the panel that you're interested in, and then go through the processing of that. Xenium was covered in detail, so I'm really not going to go through that other than to definitely say that they are releasing a 5,000 probe panel for both human and mouse in June. In fact, I was told by company reps yesterday that they will be accepting pre-orders within 2 weeks. And so I do think that they are moving from smaller scale gene panel probes to these larger scale probes. They also have an option to add custom probes on top of the ones that they have in their panels. And I think for some people that can be really beneficial. Yufei went through all of this, so I'm really not going to go through it. NanoString is the other major competitor to Xenium. They use a branched chain hybridization assay instead of a rolling circle amplification assay in terms of actually detecting the probes. They did come into some financial trouble. 10x did sue them. They did file for bankruptcy, but they were ultimately bailed out by another company. They actually just released their acquisition by Brucker Industries in Massachusetts. So I've talked to the company reps. I think they're probably going to be fine. I don't expect them to disappear anytime soon. This is their branched chain hybridization assay that they use here in order to detect each of their probes using these fluorescent markers. And again, these sequential rounds of imaging in order to detect these individual transcripts of interest. So thinking a little bit about some of the complexities of the imaging-based approaches, I think the two things people mostly talk about are sensitivity and specificity, right? Are these probes truly specific for the transcripts that they say they are? And what is the level of sensitivity? Can you really get down to the individual transcript level? These are not so easy to actually determine, and there has been a couple of recent publications that have tried to compare them directly. I think that we still don't have a great sense of which of these technologies is the best one for each of these different types of problems. But I do think that as more and more people use them, we will definitely have more information. I also think it's really relevant in the context of this conference to really think carefully about the computational approaches that each of these assays are going to need. You really cannot treat them identically. I think there are some secondary analysis that you're going to be able to do broadly across both technologies, but the sparsity of the data is going to be an issue in one versus the other. The mechanisms of sequencing-based versus imaging-based, there's a lot of things to consider, and one really does have to wonder whether individual computational approaches, especially for secondary analysis for things like niche assignment or things like that, need to actually be considered not broadly across technologies, but individually for different technologies. So I leave that to the computational people in the room. Okay, in the last 5 minutes, I just wanted to quickly highlight one of the things in my lab that we're doing in which we've utilized spatial transcriptomics, the sequencing-based, and I promise I'm not going to get too much into the weeds of the details. So I'm actually an immunologist, very interested in how immune cells function in different tissue environments. And one of the disease states that my lab uses to understand that question is allergic asthma. So allergic asthma is a disease of the lung that you get when you've inhaled too much of an allergen this time of the year with all of these pollens around. Many of us are experiencing seasonal allergies. And for some people, that actually drives an asthma disease in the lung where they have difficulty to breathe. For many patients, we know that this is primarily driven by what we call a Th2 or a type 2 cytokine response. CD4 T cells that we call Th2 cells are the primary drivers of this. And they both produce IL-13 and IL-5 in order to promote lung inflammation and pulmonary hyperreactivity. However, there's another type of T cell called the T follicular helper cell that also promotes a critical portion of allergy called IgE. IgE is critical for your sort of allergic response, like even a hypersensitivity response. And it's definitely something you're experiencing when you're having seasonal allergies. And so it turns out that these two different T cell subsets have two different functions. One is driving sort of the lung inflammation, the Th2 cells with these type 2 cytokines, and the other one is driving this antibody response, this IgE response. And through a series of information I'm not going to get into, my lab was interested in this transcriptional regulator BLIMP1, and we unexpectedly found that when we deleted BLIMP1 from T cells, specifically using a genetic system in mouse and running a mouse model that basically drives allergic asthma with chronic inhaled house dust mite, we found that although control mice, as shown here, have this beautiful infiltration into the lungs of T cells and this pink staining here in the airway, which is an indication of mucus, those mice which did not have BLIMP1 in the T cells specifically were unable to develop allergic asthma. And it turned out it was because they were unable to develop those Th2 cells, the cells that make the type 2 cytokines that actually drive the lung pathology. However, they were able to form that IgE, that antibody response, just fine. So their T follicular helper cells were totally intact and only the Th2 cells were lacking. So we were really interested in understanding how you had this one transcriptional regulator controlling one T cell type that was important for allergic asthma, but not the other. And because we know that those T cells can come from the same individual T cell to start with, the same T cell with the same T cell receptor specific for that that allergen, we began to wonder whether there was a spatial localization to this process, whether these T cells were actually in the lymph node being activated by dendritic cells presenting these allergens, and whether there was a spatial localization that underlied that distinction as to why you would get these two different outcomes. So we turned to spatial transcriptomics. This was the original Visium, using the 55 micron spots, and using cell2-loc and single cell RNA-seq data that we had paired, we were able to go back into the spatial data and identify the regions broadly, because this is not single cell, but the regions that had the most Th2 cells or the most TFH cells. And what we found was that after, over the time course of this study, from day 3 to day 5, the T cells that were the T follicular helper cells moved from the T cell B cell border zones into the B cell follicles, while the Th2 cells moved from the T cell zones into the T cell border zones, suggesting that there was in fact a spatial localization that underlied the differentiation of these two T cell populations. To really get at what might underlie that distinction, we turned to this amazing computational approach that my collaborator in the Center for Systems Immunology, Jishnu Das, developed, called SLIDE. SLIDE is a significant latent factor interaction discovery and exploration, and this is an interpretable machine learning technique for identifying significant interacting latent factors that underlie outcomes of interest using any kind of high-dimensional omic data set. Jishnu is very proud of this. It makes no assumptions regarding data generating mechanisms. It comes with theoretical guarantees regarding identifiability of the latent factors. And it outperforms at least those algorithms that are similar in nature. And so using SLIDE, we asked Jishnu to identify latent factors that underlied the spatial distinction between the Th2 cells and the, I lost my pointer here. Sorry. There we go. That underlied these two spatial localizations in the lymph node. And we were really excited to find this one factor, IL-2 receptor alpha, which we ended up going on to show was actually really critical for driving these spatial micro niches. So IL-2 receptor alpha and the IL-2 signaling pathway had previously been shown by other groups to be important for this TFH and Th2 distinction. And so we asked whether we could actually find IL-2 positive spatial micro niches in the lymph node. To do that, we used RNA-scope to actually image regions of the lymph node where we could find IL-2. And then we went into each of those regions where we could see an IL-2 positive spatial microniche and then count the number of cells around it that were also a Th2 cell with this transcription factor that we were interested in, BLIMP1, that was required for the Th2 cells. And indeed, we were able to show that all the places where we could find an IL-2 positive cell, we had an enrichment of cells that were becoming Th2 cells with that transcriptional regulator BLIMP1, suggesting that this IL-2 spatial microniche was critical for forming this BLIMP-dependent Th2 response that ultimately goes to the lung and drives allergic asthma. So I think that this kind of underscores all of the really interesting things that we can use spatial to address complex biological questions, right? This idea of cell-cell contacts, this idea of cellular neighborhoods, the idea that you have these diffusible things like cytokines that either have short-term local impacts versus things like metabolic availability that can have a longer range impact in a tissue space. And I think this is just sort of giving you some basic information that I got from a nice review, which I really recommend that you read, just thinking about how we can use spatial to address complex biological questions beyond the work that's already being done using the single cell space. And so with that, I'm just going to close and acknowledge some of the people who did the work. All the people in my lab worked on the asthma project, in particular Kun Hee, as well as my core facility. Will McDonald has really spearheaded all of the work on the spatial stuff for the core. And we've recently hired Yvonne Eisel, who's also come to build out the spatial in our core. So if anybody has any questions, I'm happy to take them. Also, feel free to ask me questions about the technologies, since that's mostly what I talked about.
So I'll start. So could you comment a bit about cost structures, because you went through all of these, just briefly. I know it's highly complicated.
So my core mostly does the sequencing stuff, so I can't really comment as well on the imaging-based things, other than to say So the imagers all cost, you have to buy a machine for all the imaging based things. And those tend to cost somewhere in the $350,000 to $450,000 range just to buy the equipment. And then of course you have to buy all the reagents and whatnot to go with it. I actually don't have a good sense of those costs just yet. The sequencing based can really vary a lot. The smaller capture sizes tend to run about $3,000 to $4,000 per sample, including the sequencing. The problem is you can't really plan a single individual sample, right? Most of those slides, if they have 2 to 4 capture areas, you have to use the whole slide. We can't do different tissues on different slides, unfortunately. So plan all your experiments in groups of 2 to 4 as needed. Yeah. And then it scales up from there per sample. Yeah.
So some of the technologies are for subcellular resolution or single molecule resolution, as you pointed out. What in your mind are good biological applications for the subcellular information? Not collapse the cell into one cell expression profile, but actually spatial distribution within cells.
Yeah, that's a great question. I mean, I'm a big lover of transcription factors. And so not all transcription factors stay in the nucleus all the time. Like one of my favorite families of transcription factors, the STAT family, they start in the cytoplasm, they get activated, and then they move to the nucleus. And I think there's also a lot of transcripts that change, right, in their localization that are not always in the nucleus. Sometimes they have to be transported in and out. So I think it would be really interesting to look at some of those types of things of when do transcription factors move to the cytoplasm. Presumably the protein would be made there, or is it always maintained in the nucleus? So I think there's a lot of interesting applications thinking about that kind of stuff. There's also isotypes, right? Sometimes you have different transcripts have different isotypes. I know the Xenium in particular is focused on being able to identify different isotypes and being able to see the subcellular resolution there. And then finally, we don't really have a great sense of microRNAs, but a lot of those are in exosomes that get exported out of cells. And so I think that's another application. Now, in that case, you'd probably have to design custom probes, but I think there's some really interesting things there. The other thing that hasn't been done yet, but I'm really excited for someone to do, is making probes for bacteria and viruses and being able to actually search for those in the context of a tissue. That might be a space where you'd be interested not just in which cell types, but the subcellular localization of the infection.